All right, let's jump right in, shall we? Today we are talking about preclinical research.
Preclinical research.
It's a crucial stage in drug development, but often overlooked.
Absolutely.
It's where scientists really figure out if a drug is safe
Right.
and if it's effective
And how effective.
before it ever even gets near a human being.
Yes, before it even gets into, like, first in human studies.
Exactly.
This is all the work that happens before then.
And we are going to be your guides
Yeah.
through this intricate process today.
All right, buckle up.
We'll uncover how researchers use a combination of lab studies,
Like in vitro.
in vitro,
In glass.
and animal study,
In vivo.
in vivo, to build that solid foundation.
So make sure that when we do get to humans, we're doing it as safely as possible.
It's like being a detective for drug molecules.
It is.
But let's start at the very beginning.
Okay.
What exactly is the purpose of preclinical research?
To gather information
Yeah.
about a drug's safety and efficacy before it's given to a human, scientists want to know, like, how does the drug behave in a living organism?
Oh, interesting.
Does it reach the target tissue? How is it broken down, eliminated? And most importantly, does it cause any harm along the way?
So it's a bit like a dress rehearsal
Yeah, exactly.
before the big opening night,
Before we get on stage,
making sure everything runs smoothly and there are no wardrobe malfunctions.
for sure.
And the first act of this dress rehearsal takes place in the lab
Right.
with in vitro testing.
In vitro, yeah, in glass.
In glass.
So we're talking about things that happen in test tubes and petri dishes.
Okay.
It allows researchers to look at the drug in a very controlled environment.
So they're basically testing the drug's potential
Yeah.
before introducing all the complexities of a living organism.
Right. Exactly.
What are some of the key tests they use at this stage?
Well, one important test is called the CACO2 permeability assay.
CACO2 permeability assay.
So permeability, we're going to talk a lot about permeability.
Okay.
This is a specific type of cell line derived from a human colon cancer cell
Interesting.
that allows us to create a model of the intestinal lining.
So it's like a mini gut in a petri dish.
Yeah, basically,
Yeah.
it's a way for researchers to see, okay, if I dose this drug orally, how well can it cross that intestinal lining and get into the bloodstream?
Oh, that's so cool.
Another one is called PAMP,
PAMP.
Parallel Artificial Membrane Permeability Assay.
Okay, I'm going to need you to break that down for me.
So it's a simplified model
Okay.
of a cell membrane
Right.
that helps researchers see, how well a drug can pass through a barrier.
Got it.
And it has to pass through barriers to reach its target in the body.
I'm sensing a theme here.
Yes.
Permeability.
Permeability is super important.
Okay.
It's all about how quickly a drug can get to where it needs to go in the body.
Okay.
So a drug with high permeability is going to be absorbed much more easily.
Interesting.
If a drug has low permeability, it's going to take a lot longer to get absorbed.
So even if a drug is effective in the lab, if it has low permeability,
Right.
it might not be so effective in the real world.
Exactly.
That's really interesting.
Which is one of the reasons why in vitro testing is so valuable.
Yeah.
It helps identify these potential problems early on.
Okay, so we've seen the drug perform
Yeah.
on a cellular level.
On a cellular level.
What's next?
After the lab, we move on to animal studies, or in vivo testing.
In vivo.
This is a really, really crucial step.
Okay.
Because it allows us to bridge that gap
Okay.
between the petri dish
Yeah.
and the complexities of a living organism.
Right, because cells in a dish can only tell us so much.
They can only tell us so much.
Right.
In vivo testing lets scientists observe how a drug behaves in a more realistic setting,
That makes sense.
and to learn about things like ADME,
ADME.
absorption, distribution, metabolism, and excretion.
Like a drug's itinerary through the body.
Yeah, exactly.
But choosing the right animal model must be crucial.
Super important.
Mm.
Different animal models have different strengths and weaknesses,
Okay.
and the choice depends on the specific drug
Huh?
and the research question.
Interesting.
So rodents are often used
Oh,
because they're small and easy to work with,
makes sense.
while larger animals like pigs or primates might be used for studies where their physiology is more similar to humans.
Interesting. Different animals for different investigations.
Yeah, exactly.
Okay, so we've established that a drug needs to get to where it's going in the body.
Yes.
But how does that actually happen?
Right.
I mean, when we swallow a pill,
Uh-huh.
it's not like it instantly dissolves in our stomach,
No, it does not.
like an Alka-Seltzer, right?
More complicated than that.
Okay.
The process of a solid drug breaking down into smaller pieces
Right.
and dissolving in a liquid is called dissolution.
Dissolution.
And only dissolved drug can be absorbed into the bloodstream.
Okay.
So dissolution is kind of like unlocking the drug's power.
Unlocking the drug's power?
And a key factor in how quickly a drug dissolves is its particle size.
Okay, now you've got my attention.
Yeah.
Particle size.
Particle size, yeah.
How does that work?
Think about it like this.
Okay.
If you want sugar to dissolve quickly in your tea,
Yeah.
do you use sugar cubes or granulated sugar?
Granulated sugar.
Granulated sugar, right. Why?
More surface area.
More surface area.
Dissolves faster.
Dissolves faster, exactly.
Okay.
The smaller the particles, the greater the surface area, and the
Hmm.
faster it dissolves.
So scientists are practically playing with microscopic building blocks
It's kind of like that, yeah.
to make sure these medicines work properly.
Right.
I never would have guessed that something as tiny as particle size could have such a huge impact.
It's really interesting, yeah.
Like, it never even crossed my mind.
And scientists even have an equation for it
Oh, really?
called the Noyes-Whitney equation,
The Noyes-Whitney equation.
which helps them understand and even predict how different factors influence dissolution.
Okay, no need to go full chemistry professor on us.
I won't, don't worry.
But give us the gist.
Yeah.
How does this equation translate to the real world?
Well, it helps us understand things like why a crushed pill works faster than a whole pill.
Oh, interesting.
Crushing it increases the surface area,
Makes sense.
which speeds up dissolution.
Okay.
And scientists can even use this equation to model how a drug will dissolve and be absorbed in the body.
So it's like a scientist's secret code for figuring out a drug's potential.
It's a tool in their toolbox for sure.
And you mentioned some real-world examples.
Yeah.
Like digoxin and griselvin.
Oh, great.
How does particle size affect them?
So digoxin is a heart medication,
Okay.
and it's a great example of how particle size can make a difference in a drug's effectiveness.
Uh-huh.
Studies have shown that if you can reduce digoxin's particle size,
Okay.
you can increase its bioavailability.
Bioavailability.
How much of that drug gets into the bloodstream
Okay.
to do its job. So in digoxin's case, smaller particles means faster dissolution,
Uh-huh.
which leads to more of the drug being absorbed
Okay.
and ultimately a better therapeutic effect.
Makes sense.
Now, griselvin is an antifungal medication,
Okay.
and it has very, very low solubility.
So it's hard for the body to absorb.
Right.
Got it.
But by reducing its particle size,
Okay.
scientists have been able to improve its dissolution and bioavailability.
So even though it's not very soluble,
Right.
they found a way to make it work better.
To make it work better, yeah.
It's like they outsmarted the drug's inherent limitations.
Exactly.
This is all incredibly fascinating.
It is.
It really highlights the incredible ingenuity and attention to detail that goes into drug development.
For sure, it's a meticulous process with many moving parts, and we've only just scratched the surface.
Really, there's more.
Oh, yeah.
This is amazing.
There's a lot more to come.
Okay, well, I can't wait to hear all about it.
You know, it's amazing how much we can learn about a drug even before it reaches a human being.
Right.
But all this preclinical research is really just the beginning
Okay.
of a much larger journey.
You're talking about the journey to getting a drug approved
Yeah.
for use in
Oh,
people, right?
Exactly.
The ultimate goal.
The ultimate goal.
Okay.
And before a drug can even be considered for human testing,
Yeah.
researchers have to submit a ton of information to the
Yes.
regulatory authorities.
Like the FDA.
Like the FDA, yeah.
Okay.
It's called an Investigational New Drug Application, or IND.
IND. Okay.
IND.
It's like a drug's resume
Yeah, it is.
before its big interview,
Before the big interview.
highlighting all its qualifications.
All of its qualifications.
Wow.
It includes a massive amount of information about the drug,
Okay.
summarizing everything that's been learned during the preclinical phase,
So like toxicology data,
toxicology data,
ADME properties,
VME properties,
dose finding studies,
dose finding studies,
manufacturing details.
manufacturing details, all of it.
Wow, that sounds pretty intense.
It's a lot.
What's the purpose of all this scrutiny?
Safety.
Safety, okay.
The regulatory agencies want to make sure that any drug entering human trials
Okay.
has a solid foundation of evidence
Right.
supporting its safety and potential effectiveness.
So they're basically the gatekeepers,
They are.
protecting the public from potentially harmful or ineffective medications.
Exactly.
Okay, makes sense.
Yeah.
So let's break down some of these key components.
Okay.
You mentioned toxicology first.
Yeah, toxicology.
What kinds of studies are done to evaluate a drug's safety?
So toxicology studies are designed to identify any potential red flags,
Okay.
any harmful effects the drug might have,
Uh-huh.
And these studies are typically conducted in animals
Okay.
using a range of doses
Oh.
and durations of treatment
To really see what's going on.
to assess the impact on different organs.
So they're basically trying to find the drug's breaking point,
Yeah, you could say that.
the dose at which it starts to cause problems.
Right. Researchers want to know how the drug behaves at different levels of exposure,
Right.
from those low doses that might be
Oh.
used in humans
Yeah.
to much higher doses to reveal potential toxicity thresholds.
So they're really trying to understand the full spectrum
At the full spectrum, yeah.
of the drug's effects.
Yes.
And what kind of signs are they looking for in these studies?
Looking for anything out of the ordinary.
Okay.
So changes in organ function,
Okay.
liver or kidney damage, effects on blood cells,
Right.
even behavioral changes
Really?
could be an indicator
Wow.
of toxicity.
So if a drug causes serious side effects in animals,
Right.
that's a pretty big red flag, right?
It's a huge red flag.
Okay.
And that's why these toxicology studies are so important. They
Yeah.
allow us to weed out those potentially dangerous compounds
Okay.
before they even get close to human testing.
Got it.
It's a big deal.
Okay, so toxicology is all about making sure a drug is safe.
Right.
But we also need to know that it can actually do its job right. That's where those ADME properties come in.
Exactly. ADME, absorption,
Yeah.
distribution,
Yeah.
metabolism,
Yeah.
and excretion.
Like a drug's travel itinerary.
Exactly.
So how do scientists actually study these properties in the preclinical phase?
They use a variety of techniques,
Okay.
sometimes even radio-labeled drug molecules,
Wow.
to track the drug's movement throughout the body.
So they can literally see where it goes.
They can see where it goes.
That's amazing.
They study how it's absorbed from the gut,
That's-
how it's distributed to different tissues,
Uh-huh.
how it's metabolized by the liver, and how it's excreted.
So they're really following the drug's every move.
They're following it, yeah.
Wow.
And this information is crucial for understanding how the drug will behave in humans,
Okay.
how long it will stay active in the body, and what might happen to it along the way.
Right. So it's not just about will it work or not.
Right,
It's
yeah.
also about understanding all the nuances of how it's interacting
All the nuances, yeah.
with the body.
Exactly.
That's fascinating.
It also helps determine the appropriate dose for human studies.
Oh, right. Because too high of a dose could be dangerous.
To be very dangerous.
And too low of a dose might not be effective.
Might not be effective at all.
So how do researchers figure out the right dose to use in humans?
Dose-finding studies.
Dose-finding studies. Okay.
In animals, testing different doses
Okay.
to find that optimal range.
To find that sweet spot
Yeah, that sweet spot.
where the drug is both effective and safe.
Exactly.
Okay.
And these studies also help determine how often the drug needs to be administered.
Oh, okay. So like
Once a day,
once a day.
twice a day, or more frequently.
Wow. So many factors to consider.
It's a lot.
And then all of this information, toxicology, ADME, dose finding,
Yeah.
gets packaged up and sent off to the regulatory agencies in the IND.
In the IND, right?
Right.
But there's more.
Oh, there's more.
We also need to understand how the drug is broken down in the body.
Oh, okay.
And that's where metabolite profiling comes in.
Metabolite profiling. Sounds a bit like forensic science.
It kind of is.
Okay.
It involves identifying and characterizing the different metabolites,
The breakdown products
the breakdown products, right?
that are formed when the drug is metabolized.
Exactly.
So it's like analyzing a drug's fingerprints
Yeah, it's like that.
after it's been processed by the body.
Right.
Yeah.
And those fingerprints
Yeah.
can tell us a lot about the drug's safety and effectiveness.
So it's not just about the drug itself,
Right.
but also about what it becomes
What it becomes in the body.
in the body.
Some metabolites might be inactive,
Okay.
while others could be active
Uh-huh.
and contribute to the drug's overall effects.
So some drugs might actually become more potent
Potent, yeah.
after they're broken down,
Exactly.
while others might produce metabolites that have unwanted side effects.
Right. So it's important to understand that whole metabolic profile.
The whole picture.
The whole picture, yeah.
Before it moves on to human testing. It's amazing how much complexity there is
There is.
in something that seems so simple, like taking a pill.
It seems so simple.
Like a whole hidden world of chemistry happening inside us.
It is. It's a fascinating process.
It really is.
And we're constantly learning more about how drugs interact with the body.
Okay, so we've talked about toxicology, ADME, dose finding, and metabolite profiling.
We have.
What else goes into the IND?
Well, we also need to make sure that the drug is stable,
Stable.
doesn't degrade over time.
Okay.
And that's where stability testing comes in.
Stability testing. Or
Researchers test the drug under various conditions.
like temperature, humidity, that kind of thing.
Temperature, humidity, light,
Okay.
to see how it holds up.
So even if a drug passes all the other tests with flying colors,
Right.
it could still be a no-go if it doesn't have good shelf life.
Exactly.
Yeah, makes sense.
Yeah, we don't want a drug that's losing potency
Right.
before it reaches the patient. And alongside stability testing, we also have early analytical development.
Early analytical development. What exactly is that?
This involves developing methods
Right.
to accurately measure the drug
Okay.
in various samples, like blood or urine.
Interesting.
And this is crucial for understanding how much drug is present
Okay.
during those preclinical studies.
So they're creating tools
Yeah, they're creating tools.
to track the drug's journey and
Exactly.
make sure it's behaving as expected.
Behaving as expected.
It's incredible how much work and detail goes into this preclinical phase.
It's a lot of work.
It's like building a case
It is.
for a drug's potential,
Yeah.
gathering all the evidence to support its advancement to human trials.
You got it. It's a rigorous process,
Yeah.
but essential for ensuring the safety and efficacy of new drugs.
Absolutely. Well, we are learning so much today
I'm glad.
about all the complexities of drug development.
Exactly.
It's really mind-boggling to think about all that goes into preclinical research.
It's a lot,
Before a drug even has the chance to be tested in humans.
right?
It's amazing.
It really highlights the dedication of all the scientists who
Yeah.
are working to bring new medicines to the world.
Absolutely.
You know, and there's one more tool
Okay.
that we use in preclinical research that we haven't talked about yet,
Okay, what's that?
and that's the biopharmaceutics classification system, or BCS.
Oh, yeah, the BCS.
The BCS.
I remember you mentioning it earlier.
I did.
Can you remind me what that's all about?
So the BCS is a way of categorizing drugs based on their solubility and permeability.
Oh.
You remember those two properties we talked about?
Yes, solubility is how well a drug dissolves.
Right.
And permeability is how easily it can pass through those cell membranes.
You got it.
And those are both really important
Hugely important
for a drug to be effective.
for a drug to work.
Okay.
So the BCS takes those two properties and uses them to classify drugs into 4 different categories.
Oh, cool. Okay, walk me through the categories.
All right, so class 1 drugs are the stars of the show.
Okay.
They have high solubility and high permeability,
By the way.
which generally means they're absorbed really well.
Okay.
Then we have class 2 drugs, which have low solubility but high permeability.
Okay.
Their absorption is often limited by how well they dissolve.
Okay.
So that's something that researchers often try to optimize.
Okay, so it's like class 1 drugs are natural-born athletes.
Yeah,
And class 2 drugs need a little bit of coaching
a little coaching, yeah,
to reach their full potential.
to get where they need to go.
Okay, what about the other 2 classes?
So class 3 drugs have high solubility,
Okay.
but low permeability.
Okay.
Their absorption is limited by their ability to cross cell membranes.
Right.
So getting them to where they need to go in the body can be a bit trickier.
Yeah, I can see that.
And finally, we have class 4 drugs,
Okay.
the problem children.
The problem children, all right.
They have low solubility and low permeability, making them very challenging to develop.
All right, so the BCS is like a quick reference guide
Yeah, it is.
that helps researchers understand a drug's absorption potential
Right.
and identify any potential challenges early on.
Exactly.
I like that.
And you know, the BCS isn't just used by researchers.
Oh, really?
Regulatory agencies also rely on it
Oh, wow.
to make decisions about bioequivalence testing.
Bioequivalence. Now, there's another term I need a refresher on.
So bioequivalence basically means that 2 different formulations of the same drug
Okay.
are absorbed into the bloodstream at the same rate and to the same extent.
So they're essentially interchangeable
Interchangeable, yeah.
from a clinical standpoint.
Exactly.
So if I take a generic version of a medication,
Right.
it should work the same way as the brand name version
Exactly, because they're bioequivalent.
because they're bioequivalent.
That's interesting.
And for those
Mm,
class 1 drugs,
the stars of the show.
the stars of the show with high solubility and permeability,
Okay.
the FDA actually allows for what's called a biowaiver. A biowaiver. What's that?
So that means they don't require those extensive clinical studies
Oh, wow.
to prove bioequivalence.
Interesting.
So in vitro dissolution testing can actually be enough.
Wow. So for these well-absorbed drugs,
Yeah.
lab tests can sometimes provide enough evidence
Enough evidence, yeah.
to show that they'll behave similarly in the body.
Right.
That seems like a huge win for drug development,
It is.
saving both time and resources,
Absolutely.
and ultimately getting those drugs to patients faster.
Which is the goal.
That's really cool.
The BCS is a great example of how scientific understanding can be used to streamline drug development and ultimately benefit patients.
Well, this has been an incredible deep dive into the world of preclinical research.
I agree.
I feel like I've gained a whole new appreciation
I'm glad.
for the complexity and importance of this stage of drug development.
It's an important stage.
It's amazing how much goes on behind the scenes
There's a lot that happens.
before a drug even gets to the point of being tested in humans.
Yeah, most people don't even realize it's happening.
It's a whole hidden world.
It is.
And for our listeners who want to continue exploring this world,
Yeah.
here's a final thought to ponder. Imagine a world where we could perfectly predict how any drug would behave in the human body,
That would be amazing.
based solely on lab and animal data.
Right.
What advancements in medicine would that unlock?
Huge advancements, I imagine.
Could this be the key to personalized medicine,
It very well could be.
tailoring treatments to each individual's unique biology?
It's an exciting possibility.
It's an exciting possibility for sure.
It is.
Until next time, keep diving deep.